We're going into uncharted territories
where no man has gone before.
We're going in the depths, in the trenches. In the trenches, bro. Where everyone else dances around these topics because they're difficult and they talk about emotions. This is getting serious, guys. We're getting into stuff nitty gritty. Very few have chartered these territories. That's why you're subscribed to me. Thank you. Make sure you subscribe if you're not. We're going to go into a crash course in oncology, and this is for people that work in clinical research or people that want to work in clinical research, and also for biotech investors. I think those two are not the same cohort, but both of those are my world lately, and there's a common language amongst both. And I think the fundamentals is not understood by neither cohort at times. I mean, it's crazy how many people work in clinical research. I've worked in clinical research for 20 years. It wasn't until year 15 I started doing or gaining oncology experience, and that was as a oncology monitor for a breast cancer study, as a contractor for a biotech company from overseas. And I was the U.S. West Coast monitor. So I monitored some academic medical centers. I monitored some private sites. And I saw that that's like oncology encompasses about half of research is oncology. And so that means that half of the industry that does clinical research doesn't know much about oncology. I was talking to a doctor today who's a pulmonologist. He didn't know what RECIST criteria was. So I'm going to try to get into, and this is by no means comprehensive. That would be like 10 hours. But this is going to be like an overview for the basics. And essentially, if you're talking about half of all clinical trials are oncology, at some point, if you stay in this industry long enough, you are going to get involved in oncology. And if you are a biotech investor, half of the opportunities, and probably the really good ones that we like to 10x our returns, are in oncology. And we can honestly use more innovation in that space. So oncology clinical trials, they rely fundamentally on standardized criteria, and it's like its own language. And there's things like TNM staging systems and the aforementioned RECIST criteria, which I'm going to get into. There's also solid tumors, liquid tumor. I'm going to try to cover as much as I can in like 30 minutes. So let's get right into it. The most commonly used staging system globally for classifying the extent of cancer is the TNM staging system. That's Tom, Nancy, Mary. And T stands for the primary tumor. So this refers to the size and extent of the main tumor. For example, if it's breast cancer, it's the size and extent of the main tumor in the breast. The higher numbers like T1 through T4, so T goes from T1 through T4, indicate larger tumor or greater local spread into nearby tissues. You do have TX, which indicates that the main tumor cannot be measured, and T0, which indicates no evidence of a primary tumor. Then you have the N in the TNM staging criteria, which is regional lymph nodes. So N stands for nodes, and it refers to the number and extent of nearby lymph nodes containing cancer cells. So N0 would be no lymph nodes spread, so no metastases. N1 and 2, it does. The reason why lymph nodes are important is because even though we don't have necessarily metastases, by having lymph nodes containing cancer cells, it's a much greater chance, much greater likelihood of a metastasis from happening. And M, like Mary, or metastases, is distant metastases. So it refers to whether the cancer has metastasized, meaning it has spread from the primary site to distant parts of the body. M0 indicates no distant metastases, while M1 confirms the presence of metastasis. So T goes T1 through 4. It also has T0 and TX. N has N0, 1, 2, and 3. And then M has either 0 or 1. So that one is binary. And while this system defines the patient's starting condition, it does not serve as a dynamic measure of treatment response. That's more for a diagnosis perspective. The distinction is vital for researchers because the baseline TNM stage dictates the standard of care comparator for the phase 3 trials, which this is why this is important, because you're going to see later in this video, in this podcast, why standard of care in oncology is super important. That's basically the benchmark for research. And for investors, the anatomical extent, so metastatic disease versus early stage T1, dictates the strategic value of an intervention. And we're going to get into why later lines of therapy are usually first and work their way backwards. So you'll hear the word adjuvant, which is treatment after surgery to prevent recurrence. So when you hear adjuvant trial for breast cancer, it means it's after the breast cancer surgery, so after the mastectomy. And this often focuses on stage 2 to 3 populations, whereas metastatic trials typically focus exclusively on stage 4. So when you hear about stage 4, you'll hear about metastatic. They basically go hand in hand. You'll also hear terms like refractory, basically the same thing. It means it's spread or it's come back, and it's come back more aggressively. So there's like an entire language in oncology that you should learn, and you're going to learn whether you're an investor or somebody that works in the space. Now, there's a difference between tumor types too. So there's solid tumors, which are the majority of cancers. Solid tumors are masses in tissues or organs and include the common cancers we know of, like breast cancer, lung cancer, prostate cancer. They're typically identified via imaging, CT or MRI, followed by a biopsy for histological confirmation. And then there's a treatment approach to those. So they often respond well to localized therapies such as surgical resection or radiation, sometimes followed by chemo, whatever the standard of care is, and depending on how early in the tumor we find it, so in the TNM staging. Now, liquid tumors are hematologic malignancies. So this is why you'll see oncologists and hematologists, right? The liquid tumors are blood cancers or liquid cancers, and they involve the blood, bone marrow, or lymphatic system. And they don't have a single solid mass. So instead, they are diagnosed. So you think of things like lymphoma, leukemia. These are diagnosed using systemic methods like blood tests, bone marrow aspirations, things like that. They do require systemic therapies, so not localized therapies, and also chemotherapy, stem cell transplants, CAR T has been one approach over the last decades that there was some promise. So there is, like, if we want to get a little more nuanced here, there are different ways to look at precision. Not all tumors are the same. Not all patients with lung cancer, for example, are identical. And this is important from an investor perspective. And this is why sometimes real-world evidence works and why sometimes it doesn't work. And there is a potential for bias when it comes to heterogeneity versus homogeneous subsets. So heterogeneity is the difference between tumors in different patients. So more critically, it's intratumor heterogeneity refers to genetic, genomic, and epigenetic differences found within a single tumor mass. And there's an internal variability, means that a single biopsy taken from one area of a tumor may miss critical mutations present elsewhere. So this is why cancer is so complicated. There's different subsets and cohorts even on the same type of cancer. For example, breast cancer, you might hear HR positive, HR negative, ER positive, ER negative, triple negative, right? These are genetic biomarkers, and they select highly specific or homogeneous patient subset or cohorts where the drug's most likely to be effective. So, for example, I worked on a trial that was for triple negative breast cancer. So all the patients in that study, not only did they have breast cancer, but they were triple negative breast cancer. Why? Because, and you see this a lot, some studies only enrolling EGFR mutations, which is a specific genetic biomarker that creates a homogeneous subset. Because the drug, the mechanism of action for the investigational product is believed to work on triple negative and not on maybe a different type of subset. And this is where the FDA has a very tough job, and lately they've been getting criticized quite a bit for not being consistent in how they approve therapies or how they approve studies, and even after they've agreed on the study design. So we'll have podcasts on this later on, but just understand that even within the same type of cancer, there are different subsets and cohorts. And if you're an investor, success in a tightly defined homogeneous patient subset is critical. So trials focused on specific subsets yield cleaner data and reduce the variability or noise inherent in heterogeneous or unselected population. This focus dramatically de-risks the drug and accelerates the path to proof of concept, which can lead to faster time to market compared to try to prove efficacy in a broad population. And this is important if you're an investor in the space. Now, there's biomarkers, there's precision oncology. We're not going to get into that too much, but just understand that there's prognostic biomarkers which predict the patient's overall outcome or natural course of the disease, regardless of the therapy used. There's predictive biomarkers which forecast the likelihood a patient will respond favorably to a specific therapeutic agent. And then there's comprehensive genomic profiling, although this particular administration, particularly the head of CBER, Vinay Prasad, argues that there's not much value add to this genomic profiling and these biomarkers. So at the end of the day, what he argues for is overall survival rate, and not even overall response rate, just overall survival and median duration of response, which we're going to get into. And also know that in these studies, just like in any other study, there's eligibility cutoffs, particularly on certain labs. So these cutoffs ensure the patient's organs and blood cell counts can tolerate the potential toxic effects of the investigational drug or the needed medication. Like, for example, in the case of tumor infiltrating lymphocytes, which is a new kind of treatment modality that's being researched, they have to use interleukin-2 to lower the body's immune response to take in the tumor infiltrating lymphocytes. So you're going to learn about absolute neutrophil counts, platelets, hemoglobins, creatinine clearance, total bilirubin, ALT, AST, and a lot more. And just understand there's going to be screen fails based on these metrics as well as based on other metrics. Now, here's what's very important for everybody. And I'm in future videos, so if you're not subscribed, make sure you're subscribed. I'm going to teach you how to analyze an investigational product and how to learn the 20% you need to know like 80% of the concepts in that particular therapeutic or mechanism of action or that particular type of cancer. So for example, if it's mucosal melanoma, we're going to look into that. And there's ways you can look at PubMed, and there's the different standard of cares and the comparison treatments and the comparator arms. But standard of care, it's what's already approved, it's what's already being used in the real world, and it's the current best practice or standard therapy. It's what is accepted by medical experts as the proper approach. And in clinical trials, the standard of care typically serves as the active control or comparator arm against what the new drug, the investigational drug, must prove superiority in phase 3 studies, which is why if you're an investor, you need to make sure you are analyzing. Don't get too excited about your phase 2 data, especially if it's open label single arm, because they could be biased, and especially with this FDA administration, they're tough on that stuff. Phase 3 typically goes against standard of care. So you got to make sure that when you're comparing one to the other, you're comparing apples to apples, not apples to oranges, or apples to another meal entirely. So the treatment lines. In advanced or metastatic settings, treatments are categorized based on prior therapy failure. So you'll have the first in-line therapy. This is the initial cancer treatment a patient receives. For example, the first line standard of care for BRAF, which is a mutant metastatic melanoma, might include a combination of immunotherapies like nivolumab plus ipilimumab. You're going to have to learn these things and learn the brand names as well, like Yervoy, Opdivo, Keytruda, things like that. Second line is if the cancer progresses or grows while on first-line therapy, or if the side effects become intolerable on first-line therapy, the next suggested treatment is second line, followed by third line, and so on and so forth. And this terminology generally applies to systemic therapy for metastatic disease. So, immunotherapy, infusions, PD-1 inhibitors, chemo, things like that. Relapsed and refractory disease. You're going to hear this a lot. The transition between treatment lines is often defined by the resistance of the disease. So refractory disease is the cancer has not responded to the initial treatment. For example, it's growing or staying the same. It seems like it's not being changed at all. In a lot of cancers, you'll see like PD-1 refractory, means it didn't respond to Keytruda, for example, which is a PD-1 blocker. I'm going to get into these mechanism of actions later. Again, this is not meant to be like comprehensive, just the basics so you understand the terminology. Relapsed disease, the cancer initially responded well to treatment and went into remission, but has since returned. So that's the difference between refractory and relapsed. Don't get them confused. I've actually seen study results from a study recently where you had a patient who started therapy. It was a second line therapy, and it took about 2 months. They got a complete response, meaning all evidence of the tumor was gone. And then 4 months later, it came back. That would be relapsed, not refractory. Refractory would be if that patient's tumor did not respond at all. So that's the difference. Now, a very, I guess, sophisticated, or maybe not sophisticated, but one of the strategies and rationales for why companies study later lines first. And I like to invest in companies that do this because it's easier burden of proof, right? So biotech companies often prioritize second line or later settings rather than challenging the first line standard of care immediately. And this strategy is driven by several factors. Number one is the unmet need and patient population. So patients progressing from first line standard of care, which is approximately 60% of patients in diseases like non-small cell lung cancer, represent a high unmet need population who receive further systemic treatment in the second line setting. You often see these second line settings third line settings ignored in some of these more niche cancers like mucosal melanoma, for example. The second reason why you go later lines first is it's a regulatory, faster regulatory speed and a lower bar. So demonstrating efficacy, which would be a strong objective response rate, we're going to get into that right now, or faster progression-free survival signal, which can be achieved faster and with a smaller trial size compared to proving superiority of a highly effective first-line standard of care. So it makes sense. And then number 3 is the proof of concept. A strong proof of concept in the second-line setting rapidly justifies the massive investment required to eventually challenge the first-line standard of care in a larger phase 3 trial. And it doesn't mean patients in the second line are harder to treat necessarily. Like you don't know which one's in the first line. Like every patient who's in the second line at one point was in the first line. So it's not exactly accurate to say patients are easier to treat in the first line, although you do get more responders, but it doesn't mean that just because something works in the first line means it will work in the second line, and vice versa as well. And this is why we got to do the studies. And so the oncology clinical trial life cycle, there's phase 0, which is not a mandatory step in the drug development process. These are gathering very early PK or PD, so which is pharmacokinetic or pharmacodynamic data, to see if it's worth pursuing. Sponsors can omit those if they already have sufficient preclinical data, or if the compound uses materials for which the toxicity data is already known. They're going to have these conversations with the FDA. Now, combining phases you can do. It's very frequent you see a phase 1/phase 2 study. Phase 1 to assess efficacy at the recommended phase 2 dose. Where it gets interesting is the phase 2 and the phase 3. So essentially in the phase 2, what you're looking for are initial efficacy signals and go, no-go decisions. So when you're in a phase 2 study, the main goal is to determine if the treatment will demonstrate sufficient activity against specific types of cancer in a smaller group of patients, usually 25 to 100. A common design is the Simon 2-stage model, and this involves treating a small initial cohort and setting a minimum response threshold. If the response rate is below the threshold, the trial is stopped due to futility, saving significant resources because it is expensive to do these studies. And this is why we have interim data analysis and things like that. This is why entering data on time and answering queries in a timely manner for research sites is so important. And then the endpoints in the phase 2 study are objective response rate and progression-free survival. Now, what we really want to see is overall survival and improved quality of life, but we can't actually get that until phase 3. And so what we're looking for as far as the endpoints of most of these cancer oncology phase 2 studies are ORR, objective response rate, and progression-free survival, and determine if the result is statistically significant, often defined as what you'll hear as a p-value of less than 0.05. Now in phase 3, they want to see superiority over the established standard of care. So these are large-scale randomized clinical trials. And by the way, we're going to get to how these are approved, fast-track designations, and things like that as well. But just bear with me a little bit. I know you investors are anxious for that kind of stuff. So when the phase 3 trials, they're really the pivotal trials. The endpoints must demonstrate what I said already, overall survival. They're usually tested against the standard of care. It'd be nice to see standard of care being tested as well in phase 2 or double-blind randomized, which is the gold standard. You can't do placebo-controlled. In oncology, it's unethical, but you can do standard of care. as the comparator. And there's a stat, like approximately 90% of successful phase 3 oncology trials ultimately lead to FDA approval. So this is why these are called pivotal trials. 90% of successful phase 3 trials get FDA approval. This is why stocks jump so much on phase 2 data. But the phase 2 data doesn't mean the phase 3 data is going to look good. So sometimes you'll get like a very expensive stock with good phase 2 results that don't perform well in phase 3, especially when they're not double blind, or at least double arm. They have a comparator arm. So those single arm studies, I'm a little cautious about on phase 2. But it doesn't mean it's not going to work in phase 3 either. Everything there's a context and a rationale for everything. And then you have phase 4 studies. So these occur after regulatory approval and are designed to collect more information on long-term safety. You'll often hear that confirmatory trials, these are required for drugs approved under the accelerated approval pathway. So here are the trial endpoints for the most part. There's more, but you'll see a lot of these. I already talked about overall survival, and it's time from treatment initiation until death for any cause. Progression-free survival, time from treatment initiation until objective disease progression based on imaging criteria. Objective response rate, percentage of patients achieving complete response or partial response. The other factors are stable disease or progressive disease. And then you have duration of response, which you'll see duration of response in phase 2 studies too. Median duration of response. It's basically like the overall survival for phase 2, because you can't do overall survival, but how long does the tumor actually continue to respond to treatment without cancer growth or spread? And objective response rate provides the initial signal that a tumor shrank. Duration of response provides the critical measure of durability. It's probably, in my opinion, more important because you're starting to get into things like money. And if you read Vinay Prasad's book, he's now the head of CBER, which is in charge of oncology regulations. He talks about, well, what good is objective response rate if it doesn't actually last, is expensive, and the patient doesn't live much longer than they would on the cheaper standard of care? And he's got a point. I mean, tumor shrinking is fine, but ultimately what we're looking at is, are you going to live longer, and are you going to have better quality of life while doing the treatment? And so median duration of response is kind of like a secondary endpoint or a derivative of overall survival. It provides the durability. So how long can we delay disease progression? And this now gets into like the financial benefits of what it costs, what it's worth paying, what do payers, and this is especially for the investors, important stuff here. So just because you have great overall response rate doesn't really mean much in this administration if you don't have durability of response. And for example, standard of care immunotherapy drugs like Keytruda, also known as Pembro, exemplify the value of durable response. In non-small cell lung cancer, patients with high PD-L1 expressions, which is about more than 50% of them, phase 3 trials, Keytruda significantly extended survival compared to chemo. They had an median overall survival of 26.3 months versus 13.4, so basically doubled the overall survival, which is adding a little more than a year to someone's life. And these are considered substantial survival gains, and a direct evidence of a highly durable clinical response. Keytruda was like the world's best-selling drug until I think Ozempic or Wegovy, one of those GLP-1s, took over. So whatever indication you're looking at in oncology, you got to understand what's the best standard of treatment and what is the overall survival for that. And then you got to kind of look at how it did in phase 2 as well and compare it to whatever you're invested in. So like for melanoma, in a phase 3 comparison against another immunotherapy, ipilimumab, Keytruda demonstrated significantly longer progression control, with median progression-free survival extended to 8.4 months in the Keytruda arms compared to 3.4 months in ipilimumab. And when you're analyzing yours, you got to look at what is the Keytruda for your indication. It might be Keytruda itself. I'm not going to get into hazard ratios because this would be a way too long of a video, but I need you to look into hazard ratios because essentially for phase 3 trials, especially, success is defined by comparing the treatment group to the control group using a hazard ratio, which measures the instantaneous risk of an event like progression or death. So a hazard ratio of 1 means the drug offers no benefit over the control group, whereas a hazard ratio of 0.75 means the drug lowers the risk of progression or death by 25%. So you guessed it, a trial is successful if it achieves a statistically significant HR, typically less than 1, for the primary endpoint, progression-free survival or overall survival. Interim analysis, you're going to see this all the time. They always employ interim analysis, phase 2, phase 3, and they have think of it as a sneak peek of the data. And the trial may be stopped early if the result is overwhelmingly positive. So this is called the efficacy stop, and you'll see the stock prices like soaring. They typically require a high statistical bar, like a p-value of less than 0.001 instead of a 0.05. Or if the result is clearly failing, the stock will drop significantly, typically p of greater than 0.5. We're not getting into all that. What is very important for you to understand is RECIST, RECIST, and it's the universally adopted standard for objectively measuring changes in tumor burden in solid tumors only, and it relies on measurements of specific target lesions. So just Google RECIST, but the gold for the investors out here, so you're going to have complete response, which means disappearance of all target lesions, basically cured. Although I've seen complete responses come back in the past. Partial response, at least a 30% decrease in the tumor size compared to the baseline measurement. You see this a lot. Progressive disease means it's progressing at least a 20% increase. Now, beyond the complete response, partial response, investors track the depth of response, meaning the median percentage shrink across all responding patients. So a greater depth of response, like 60% shrinkage versus 30% shrinkage, is empirically associated with a significantly more durable response. So that's another sign of durability in this sense. Now, there's a lot more with RECIST you can get into later. There is toxicity grading. So just like efficacy is important, we got to look at toxicity because some patients can't tolerate these treatments. No matter how good it is, if they can't tolerate, it's not good. So there's basically a standardized reporting of adverse events, and you're going to use the National Cancer Institute's Common Terminology Criteria for Adverse Events, CTCAE version 5 or 6, and it assigns a specific severity grade to each adverse event term. So mild, moderate, severe, life-threatening, or death. And just know that you're going to look at those. You're going to look at serious adverse events because sponsors are looking at that too. And just like they're comparing with the standard of therapy for efficacy, they are also as far as safety. And so for some investors, the safety thresholds or the red flags are more than 2 treatment-related grade 5 death events. and that has to be treatment related. So people in oncology unfortunately die, but if the investigators deem it's related possibly to the drug, that's more than 2 of those in a study, it's a red flag, or a high incidence of severe toxicity, typically defined as greater than 30% of patients experiencing grade 3 or grade 4 treatment-related adverse event. Now, you're going to hear things like FDA expedited programs, fast track, breakthrough, priority review. Let's go through some of those right now. So, fast track designation is intended for therapies that fill an unmet medical need. The primary benefit is enhanced communication with the FDA, which is called a rolling review, which allows questions and issues to be resolved quickly, often leading to earlier drug approval. To qualify, a drug must demonstrate a potential advantage over existing therapies. So typically phase 2, then you can get a fast track. Breakthrough therapy designation, it's different, is granted when preliminary clinical evidence indicates that the drug may demonstrate substantial improvement over available therapies on clinically significant endpoints. So breakthrough designation grants all the benefits of fast track plus guidance from the FDA on trial design. Breakthrough therapy designation is considered regulatory rocket fuel. And then you have priority review, which is a designation given a drug application that is intended to treat a serious condition and offers the potential for significant improvement in safety or effectiveness. And it makes the FDA commit within 6 months to review the application, compared to the standard review of 10 months. Now, there's a strategic value of priority review. So distinct from priority review designation is the priority review voucher. And this is a transferable financial instrument. Like, companies can actually sell this. So don't get priority review designation mixed up with priority review voucher. If a company successfully registers a product qualifying for a priority review voucher, it is granted a voucher that can be redeemed or sold to another company. Redeeming the voucher grants a priority review, the 6-month timeline for any subsequent product chosen by the voucher holder. This potential for a 6-month earlier market entry for higher revenue pharmaceutical products, with market values currently around $100 million for this priority review voucher. I've invested in companies that have had priority review vouchers that have sold them in the past. And then there's a brand new one. This new FDA is trying to shake things up for better or for worse. It's called the Commissioner's National Priority Voucher, which it's a pilot program right now, which offers an opportunity to reduce the review time for certain applications from the typical 8 to 12 months down to just 1 to 2 months using a collaborative Tumor board-style review process, so you might hear that. Now, accelerated approval and confirmatory trials. Accelerated approval is a crucial strategic tool that permits the FDA to approve drugs for serious conditions based on a surrogate endpoint, like ORR, which is objective response rate, or PFS. Now, these are not the gold standard endpoint like overall survival, but these are the ones you can get early on in a phase 2 study. And it's reasonably likely to predict a true clinical benefit. This is called a conditional approval. But under the accelerated approval pathway, it's conditional. So they can approve the drug conditionally, but to verify the clinical benefit suggested by the surrogate endpoint, the sponsor must conduct post-marketing confirmatory trials or phase 3 trials to demonstrate overall survival. So that's really the name of the game. Then there's conditional approval risk, means that the long-term valuation of a drug approved via accelerated approval is perpetually contingent on the success of the post-marketing trial. So there's a lot to look into just those pathways. Like, I can do an hour on those. We're not going to do that. There are some operational and statistical red flags that I think are important to discuss. So for biotech investors, analyzing the protocol and the early data reveals clinical red flags that signal high operational or regulatory risk. So low initial efficacy. Any ORR below 15% in a non-selected phase 2 population often indicates the drug is a dead asset, unlikely to succeed in a costly phase 3 study. If you have a high screen fail rate, especially in oncology, high screen fail rates are common in all studies, but in oncology, if it's greater than 50%, it suggests the eligibility criteria are too strict or the patient population is too rare, which will lead to long enrollment delays. No control arm. This is the part, especially this new FDA, they're going to have a problem with no control arm. I think when you get to a phase 3, having a comparator, if you use something like physician's choice, it's hard to compare. So it leads to noisy, unreliable data. It's not going to satisfy these regulatory bodies right now. And then single arm phase 3, those are okay on phase 2. But on phase 3, if you don't have a non-randomized single arm phase 3, unless it's like justified for a rare disease, is highly problematic. And then safety run-in, toxicity, like I said earlier. So overall, I think these are the things you got to look at from a oncology perspective. I think there's a huge world out there. Just to summarize, TNM, T is how big is the original size, and did it jump a lymph node? M, did it spread to distant organs? First in line, why do biotechs go for second line first? Because first in line standard of care, like Keytruda, is really hard to beat. Second line, because 60% fail in the first line, it's a huge unmet need. And the control arm is like chemo, although now there's other better control arms, but it's an easier bar. And it's a little faster. Second line to bridge to first line, so always work your way from the later lines down. Understand the labs, so the ANC, platelets, hemoglobins, especially if you work in the space. RECIST, understand the response evaluation criteria in solid tumors. And you pick up to 5 lesions, maximum is 2 per organ, and you measure longest diameter of each, add them up, sum of longest diameters, and then the responses. Complete response, all target lesions gone. Partial response, the diameter, sum of longest diameter shrinks more than 30%. Progressive disease, it means the sum of longest diameter grows more than 20% or a new lesion. And then stable disease is everything else. So the AE versus SAE versus SUSAR versus the safety versus the toxicity, all of these play a role into will the FDA approve it or not. And they have the fast track, which is the rolling review, frequent FDA meetings, saves about a year. Breakthrough designation is all of the above, plus intensive guidance, but you need preliminary clinical evidence, like 50% ORR, when standard of care is 10%. That, not necessarily those numbers, but significant preliminary clinical evidence. And then accelerated approval, which they'll say, okay, we'll approve it conditionally, but we need to see the overall survival on the phase 3 to prove the overall survival benefit. They're going to have 180-day progress reports. And if it fails, the FDA yanks approval. 7, drug withdrawn from 2021 to 2024. So just some simple investor math. Accelerated approval is 2 to 3 years early revenue. Confirmatory failure is 100% value wipeout for the most part. Hopefully there's more indications. And that's about it. Again, there's a whole lot more. I'm going to get into specifics like breast cancer, non-small cell lung cancer, and even in those, like I discussed, you're going to have homogeneous cohorts. You're going to have cohorts that are different from even each other, right? Based on biomarkers, based on some tumor heterogeneity. It gets complicated. I just wanted to keep this one pretty simple. Let me know what you think. Hopefully it helped. Like, subscribe, comment, share, and I'll catch you later. Bye-bye. Hey, if you guys like that kind of content, make sure you like, subscribe, comment, share. Stay tuned for more videos.